Good evening, everyone. Trust you are all doing well and are taking good care of yourselves and your near and dear ones. Welcome to the third session of Scientifically Speaking Season 4. This is a public engagement series that aims to connect high school students and anyone who's keen to learn with interdisciplinary scientists trying to solve complex problems. My name is Yukti and I'll be your host for today's session. I lead the academic communications work here at Ashoka University. Prior to joining Ashoka, I finished my PhD in chemistry from Tata Institute of Fundamental Research, Mumbai. So without much ado, I will quickly introduce our speaker for today. We have with us Dr. Basudev Maji. He is the Assistant Professor of Chemistry and Biology at Ashoka University. Basudev earned his PhD from the Indian Institute of Sciences, Bengaluru, and subsequently moved to Harvard Medical School and MIT to pursue his postdoctoral studies. His research group works on synthetic biology and chemogenetics to address various genetic and infectious diseases. His work primarily involves protein engineering and chemical biology methods to sort of understand biology underlying various diseases and their chemopreventive development. As we have been communicating through the social media as well as through the registration portal that Dr. Vasudev will be discussing about how we can harbor some of these microbial weapons to improve human health. He will be in detail talking about how viruses are being used to treat various bacterial infections. He will also discuss an extremely powerful technique that also bagged a Nobel Prize in chemistry. It is known as the CRISPR-Cas9 that scientists developed to edit an organism's DNA. This technique was sort of adopted from a naturally occurring gene editing system that the bacteria uses as an immune defense system against a viral infection. Using CRISPR technology, scientists have gained the ability to sort of delete the undesirable traits and also potentially add desirable traits with precision. So this powerful technique is being used to treat, for example, mice with genetic diseases such as deafness, sickle cell anemia, etc. And it is anticipated that in the near future, it could also be used to treat humans with similar diseases. So before we begin, I would request our audience to please drop in your questions during the session in the Q&A box, and we'll take them up in the question and answers session after Basudev finishes his presentation. On that note, I would like to invite Professor Maji. We are very excited and we look forward to your session. Over to you, Basudev.
Thank you so much, Yukti, for the kind introduction. Welcome you all. I hope you all are doing great. And good evening to all of you. So before, I think I should start with sharing my screen. All right. So screen, I'll just
perfect.
Done by laser pointer. Okay. All right. Should be good. Great. Again, welcome you all. So today, the topic of today's discussion is microscopic warfare and human health and their potential application in clinic. So as Yukti already mentioned, I'm going to talk about a lot about microbial microbes, and that divided into several parts. The outline is like that. I'll give brief introduction on the microscopic organism and microscopic warfare. Then I'll talk about antimicrobial resistance, which we are facing a severe problem with. And then we'll also move to microscopic warfare and their potential clinical prospects and applications. Then I'll touch upon microbial defense mechanism and how we can formulate that and take it forward to the clinic. Also, I'll be talking about another microorganism, that is a virus, human virus, and I'll talk about how to transform this kind of virus, human virus, and take it to therapy. At the end, I'll give you a glimpse of research that we do at Ashoka, and with that, I'll conclude my talk. Okay, so when you talk about first of all, we all must be quite familiar with this proverb that survival of the fittest. And in the way of survival for the fittest, a defense is the integral part of it, and base defense is the offense. Now, if you think about this kind of fight, it can be at different scale. it can start with a planetary system, for example, a defense system against asteroids. Or if you scale it down further, it can be anti-missile system at a smaller scale, right? Now, if you further trim it down, it can be a personal defense system. It can be our defense, our own defense against micro, for example, bacteria, so as for virus. If you further scale it down to the molecular level, then we need to have a defense mechanism against, for example, molecular toxic molecules. For example, the gas, tear gas, or sorry, the mustard gas that was used in the era of World War. You can further trim it down, and you can get to at the nuclear level. And at the end, it can be the other end of the spectrum is electrical spark that some of the creatures like eel uses for its defense mechanism. Now you can understand the defense can be at a very broad spectrum in terms of its dimensions. Now today in this topic, we're gonna only focus on microscopic warfare and defense, mainly involving bacteria and viruses. Now both bacteria and viruses, they can be, I need to remove the top of this page. Okay. I hope the title is visible to all of you. Can you please confirm?
Yes, Vasudev, we can see.
Okay, all right. So now when you talk about bacteria and viruses, and both of them can pose serious life threat. For example, bacterial infection can lead to different high temperature, and it can also activate our immune system that can lead to serious diseases. At the same time, as we all know, COVID has taught a lesson to us that viral infection can be dangerous. So both bacterial and viral infection can be life-threatening. Now, like COVID virus infection, bacterial infection, especially the resistant bacteria, can develop the next potential crisis. So in this field, scientists are working for a very long time. So for antimicrobial therapy, so scientists are able to formulate and provide multidimensional strategy. For example, developing drug molecules targeting cell wall biosynthesis of bacteria, or targets different biomolecular synthesis, for example, DNA, RNA, and protein, or very crucial metabolites, for example, folate synthesis. So scientists have targeted all these different pathways in pathogenic bacteria to kill them. However, these bacteria, they are quite smart and they can evolve with time. And many of these bacteria, they can evolve and gain resistance against many of these drug molecules. As you can see, it's listed on the right side. All these molecules, all these drug molecules, they become inactive in some of the resistant bacteria, and we call them superbug. Now, the superbug can pose a serious challenge, and that can turn into epidemic and pandemic. Now, at the same time, you might wonder that we already have a lot of antibiotics in the market. Then why do we need other different strategies, and why should it be scared? Because antibiotic molecules, they do a lot of side effects. They come with a cost. They can give you a lot of side effects. For example, nausea, diarrhea, stomach problem, vomiting. Not only that, it can lead to serious problems. For example, ototoxicity and nephrotoxicity. These are quite irreversible, especially in elderly people. Now, that's the reason we need to have an alternative strategy. To take an alternative strategy, wouldn't it be great if we can have a magical cure without much side effect? Obviously, right? Now, we all have heard about the holiness of Ganga water, right? Ganga Jal. But why so? Why do you consider Ganga Jal as holy water? And you must have noticed that across India, people use and they sparkle Ganga Jal in different auspicious occasions. Then can it have any beneficial activity? For example, beneficial therapeutic activity, medical activity. To find out that answer, to look into the holiness of Ganga water, a team of scientists, medical practitioners from Department of Phage Therapy, Uttarakhand as well as from Maharashtra. They did this study to look into the effect of Ganga water on psoriasis infection. Now, as you can see in this figure, so after applying Ganga gel in certain formulation, in medical formulation, they're able to find impressive development in patient condition. For example, you can see some of these conditions were drastically improved, and it was quite impressive. You can see on the right side as well, after applying Ganga gel in a certain formulation, doctors are able to cure some of the diseases in some of the patients. I won't claim that in all the patients, in some of the patients. Then the question comes, is it real? Is it scientific? Is it true? To look into the scientific validation of this kind of medical observation, then we have to go back to the microscopic level. So as I have already mentioned, bacteria can infect humans, and that can cause different diseases. Now, not only that, bacteria are also not trouble-free. They can have their own predators. For example, even smaller particle like virus, we call them bacteriophage or phage virus. They can infect these bacteria and they can kill them. So then the question is, do we have this kind of particle, or we call bacteriophage, in any of the sample? That means the Ganga jar. So before that, let's understand how it really works in scientific work. For example, here you are seeing a bacteriophage that can infect its prey bacteria. So this is the bacteria over here. Once it gets attached to the bacteria, then it can inject its genetic material, and that genetic material is going to hijack the system, bacterial different machinery. For example, protein synthesis machinery, DNA and RNA synthesis machinery, and it's going to make tons of its own nucleic acid, that means DNA and RNA and protein, then reconstitute into thousands and thousands of new virus particles, and then it's going to burst it out and lyse it, and then go after another host cell, that means bacteria. So this is possible. Now, not only that, there are several reports in literature, especially during World War II. So it goes back to the history of development or identification of phage in 1915 by British bacteriologists. And immediately after identifying bacteriophage, that means the virus for bacteria, then it started rolling out and started becoming popular in the medicine. So some of the countries, for example, that time Soviet Union and some of the European countries, for example, Georgia, Poland, and all, they started investing time on developing bacteriophage therapy. And especially in Soviet Union history during World War, there are several reports that they applied bacteriophage therapy to the infected soldiers in the battlefield, and it was able to cure thousands of soldiers. So it was effective. However, there was a sudden turn. In 1928, Alexander Fleming discovered the first ever potent antibiotic drug molecule, penicillin, and that changed the direction of especially the Western world. They got more interested into penicillin drug, and then the focus was entirely shifted to this drug molecule, especially in the Western world. However, countries like Russia and Poland and some of the Eastern European countries, they kept bacteriophage therapy as an alternative to conventional drug molecules, especially antibiotic drug molecules. But even after developing drug molecule like penicillin, the problem was not solved because of the abuse and overuse of antibiotics, especially in patient as well as in agriculture and farm. Certain strains of bacteria started developing, and they become resistant to some of these potent drug molecules or antibiotic drug molecules. That started giving an alarm, or it started giving an indication that antibiotic drug molecule is not going to be enough in the long term. Not only that, UN has already estimated that if this continues, if we cannot solve the problem of microbial resistance, antibiotic microbial resistance, that means AMR, by 2050, there could be 10 millions of deaths every year. So this is a serious challenge that we have now. So to find an alternative strategy, researchers started looking back, looking back into the bacteriophage therapy again. So recently, there is a study in the US, in the UCSD, University of California, San Diego, and there they did a thorough investigation on bacteriophage therapy, a systematic study where they took selected 10 patients and then treated them with bacteriophage, and in some of the cases it was combined in the combination with drug molecule, and then they looked at the outcome of the therapy. So as you can see in this diagram, as you can see, the blue part, so in total they had 10 patients, and out of that, 7 of them showed positive outcome. They're able to cure 7 patients out of 10 patients. That means 70%. It's tremendous. It's not a joke. And all these patients, they had different kind of deadly multi-drug resistant bacteria, for example, Staph aureus, Pseudomonas aeruginosa, E. coli, and so on and so forth. So phage therapy is potent. It can do a lot of, it can give positive, it can bring positive outcome. Then the question comes, is that going to be enough? Probably not. We still need to work on the therapy and increase its efficacy. And some of the times it might require a combination with drug. In the parallel, with the development of phage therapy, in parallel, there was another avenue which was taking a shape over time, which is called microbial adaptive immunity. So as you can see, earlier I have mentioned that these bacteriophages can infect bacteria and kill them, right? Now, there cannot be an unidirectional selection pressure on bacteria by virus, right? Bacteria has to fight back, otherwise they'll lose the battle. And by this time, if they do not have any defense mechanism, by this time they should be wiped out. That means it should lead to the evolutionary dead end, which is not the case, right? We still have bacteria as well as virus. So scientists wondered that is there any defense mechanism that is present inside this bacteria, which is protecting them from these infecting viruses or phages. For example, if you take just a single drop of seawater, you can find a million of bacteria and 10 times more viruses and some other smaller organisms. Now, as you can see, this bacteria and viruses, they are coexisting. Imagine if the virus has an upper hand, then that's going to kill all the bacteria, but it's not happening, right? They're coexisting. That means they have a defense mechanism to fight against each other. To look into that, to dig it out, it goes back to 1980s when Francisco Mojica, a scientist from Spain, he was looking into the genetics of certain bacteria and archaea which can survive in extreme conditions. These are called extremophiles. When he was looking into the gene sequence of some of these bacteria and archaea, he was able to find distinct patterns in the genome. For example, these elements that you are seeing in orange color, these are called repeats. That means these are the same gene sequence over and over. So they are repeated. These are called repeats. At the same time, they are separated by unique DNA sequence, the one in black, green, blue, and so on and so forth. So basically, distinct, unique DNA sequences are separated by repeats. So scientists started wondering why these bacteria have this kind of repeats. So first of all, Francisco and Ruth Johnson, they termed this kind of repeats in bacterial genome as CRISPR. That means clustered, regularly interspaced, short palindromic repeats. Later on, Dasco found that these spacer sequences, that means the unique sequences, they're not part of bacteria or archaea. They're from outside, extra chromosomal. And he also found that they have significant similarity with phages, bacteriophages. Then he started drawing a conclusion that there must be some relation between this phage infection and these spacer sequences. Later on, Dasco found that these spacers, might be involved in the defense mechanism of these organisms, that means bacteria and archaea, against phages, bacteriophages. For example, we need a lot of bacteria in dairy industry to make dahi, yogurt, or cheese, right? Now, these bacteria, these good bacteria, they can be infected with viruses, right? And that could be a huge loss for the dairy industry. So scientists were thinking how to make these good bacteria immune to the virus. That means how to vaccinate them. So to answer that question, two scientists started working on it, Barangu and Howard. What they did, they incorporated a segment of phage virus DNA into the bacteriophage DNA into bacterium, that means these good bacteria, and found that these bacterium become resistant. So that was wonderful. That was a huge discovery for the dairy industry. But scientists had no clue how it works. So then scientists started putting together efforts to solve this mystery. And 2016, these are the scientists, they are gathered together in a meeting, and they recognized for their significant contribution to the field of CRISPR to solve the molecular mechanism, how this protective mechanism in prokaryotic bacteria takes place. So in this meeting, I was fortunate enough to meet these two Nobel laureates. As you might know, Doudna and Charpentier, they were awarded with Nobel Prize for the discovery of gene editing methods. So then, in this landmark paper that came out in 2012 in Science, these two scientists, Doudna and Charpentier, they solved all those problems, all the puzzles that are taking place inside this prokaryotic bacteria. For example, when bacteriophage, that means the virus, infects the bacteria, how it processes the invading genetic material and then stores some of the part of this invaded genetic material into its genome and makes spacers. So these are unique molecules, unique DNA sequences that are stored in the form of spacer. And then later on, these immune bacteria, they can process the stored genetic material, that means the spacer, in the form of RNA. This RNA is going to be processed further, and then it can bind to other proteins, for example, Cas protein. This Cas protein and this RNA is going to bind with each other and reconstitute DNA nuclease. What that means? DNA nuclease, that means it can cleave DNA. And then this bacterium, it can use this reconstituted RNA and protein complex in its defense mechanism. How? In the second step, in the later on, when the similar kind of phages or viruses try to infect this bacterium, which is already immune, it can utilize this RNA protein molecule to target the infected genetic material coming from the viruses and degrade them. That is how this bacterium is not immune to this particular virus. All these steps and molecular mechanisms solved, and that's the reason both Doudna and Charpentier were awarded with Nobel Prize. The mechanism is like this: there are two fragments of RNA, and they are able to engineer and make a single RNA, which was known as sgRNA, and then that binds with an enzyme known as Cas9, and this is the active endonuclease. Now it can target DNA and then cleave it. So this is the first ever in vitro gene editing data that Doudna and Charpentier produced. So as you can see, this is the original DNA, and in the presence of this protein and RNA complex, it can cleave, it can give smaller fragments. This is how bacterium also uses to disintegrate or to degrade the infected genetic material coming from viruses and protect them. So immediately after the discovery of and deciphering of the molecular mechanism of CRISPR, then other scientists, for example, George Church and Fan Zhang, they modified the system to make it compatible with human cells, mammalian cells, so that we can take this technology towards the clinic. Not only that, David Liu started developing a parallel avenue known as base editing to induce very precise and very precise molecular changes or DNA changes in the genome of mammalian and mammalian cells and in the organism. So here is a nice video coming. It's nice clip from Nature Group, which explains how this molecular machinery or seizure works. For example, this Cas9, which you are seeing in blue color, that binds with the red molecule known as sgRNA, that means single guide RNA, and then reconstitutes or forms endonuclease, DNA endonuclease. That means it can target DNA. How? To target DNA, it looks for a very specific sequence in the first place. For example, postal code that we use to deliver letters, right, or parcel. Once it finds this postal code known as PAM, then it uses its guide RNA spacer, and then it unwinds the double-stranded DNA, targeted double-stranded DNA, and one of the strand is going to bind to the guide RNA, and then it forms guide RNA and DNA duplex. After that, once this sequence matches, then the enzyme activates its nuclease domain, protein domain that can induce nuclease. You can see the two cards. So basically, after matching the sequence, it activates itself and can induce double-stranded break in the target DNA. So this is the way it works. As you can see, it's very sequence specific, and it entirely depends on the guide RNA sequence of this protein, which is known as spacer. That means the sequence has to match the target sequence, and once the complementarity matches, then the enzyme is going to induce a double-stranded break. So that means, let's say if you have to target different region in the genome, then what you need to do, you can use the same Cas9. That means the seizure. That's the molecular seizure. Only thing that you need to change is the sequence of this part of the guide RNA. That means the spacer. So you can attach or you can design different guide RNA sequence depending upon the target sequence, and they can identify their cognate target sequence and induce a double-stranded break. So with this technology, virtually it becomes possible to target any genomic locus in the genome, and it was virtually possible to do gene-specific manipulation, precise manipulation. With this technology, it was so diverse and versatile and powerful that there are several disruptive technologies were evolved. For example, people have used CRISPR-Cas9 method in disrupting a functional gene. For example, let's say if this is an oncogene, then you can disrupt it to get anti-cancer activity. At the same time, one can delete this kind of faulty gene. Not only that, you can also insert a foreign gene which is not present in the organism. You can also do genetic regulation as well as transcription regulation at the same time. You can use CRISPR-Cas9 system to induce a precise single base resolution chain known as base editing. At the same time, people have utilized this CRISPR-Cas9 system for the sequence-specific recognition for the application in disease diagnostic and detection. So all these kind of hundreds of different disruptive technologies have been developed based on CRISPR system. Now, I'm going to take you through some of these applications. The first one is a CRISPR-based gene therapy to solve hypercholesterolemia. So there was a beautiful survey, genetic survey, happening in the US, in Texas, between two groups of people, American European and American African. People are able to found that there are two nonsense codons which disrupt the activity of this particular protein known as PCSK9. And once they tend to have low cholesterol level. And now, there are also certain mutations which can activate the degradation of PCSK9 with the association of low density cholesterol or low density lipid receptors. So once they bind, they can be degraded inside cell. Now, if you do not have this receptor, then cholesterol and other low density lipids are going to floating around, and that can lead to hypercholesterolemia. So to develop a strategy to solve this hypercholesterolemia problem, people have used CRISPR targeting PCSK9, and they're able to disrupt the function of PCSK9. As you can see, this is the level of protein in before treatment, and after treatment, the level of protein is significantly dropped down. Not only that, there was almost like 40% reduction in the cholesterol level in these treated mice. And another example, it was also able to solve muscular dystrophy, a muscle disease, where you can see the progressive generation of muscular disease leading to the bedridden condition for some of the patients. And one of the reasons could be deletion of an exon called exon 44 in dystrophin gene. And once this is deleted, exon number 44 is deleted, it can lead to the generation of stop codon at exon number 45, and that stops the expression of the dystrophin gene. You can see there is no red signal. That means dystrophin protein is not getting expressed. However, with the help of CRISPR, people were able to restore the expression level of dystrophin gene, and that was able to solve some of these dystrophin genetic problems. Another problem is beta thalassemia, where a single base resolution mutation can lead to the aggregation of hemoglobin, leading to the crescent-shaped RBC, which won't be capable of carrying oxygen. In this condition, it leads to the thalassemia disease state. People were able to utilize CRISPR in solving the problem. As you can see, after applying CRISPR, it's the sickle-shaped RBCs, they took a round shape, that means the wild type, that's the normal shape. So people are able to utilize CRISPR technology in solving thalassemia problem as well. Here you can see people are able to use base editing method based on CRISPR to solve hypercholesterolemia. They're able to reduce 61% blood cholesterol in human non-human primates, and that is under clinical trial. Not only genetic diseases, CRISPR is also used in detection. For example, there is a method called Sherlock, which can detect RNA base, and then the CRISPR enzyme becomes activated, and through their bilateral or bystander activity, we can amplify the signal to detect genetic diseases or molecular detection like DNA and RNA. So I won't be able to cover all the applications. In this slide, I'm going to give you some more idea about the application and the broadness of this method. For example, in normal Mendelian inheritance, you would expect that there will be 50% gene transmittance from the parents, right? And that's how we maintain the diversity in the population. However, one can apply CRISPR generating engineered mosquito, and that can lead to generation of both similar kind of sex. That means the gender, and eventually that can lead to same gender population, and that is going to force the population to shrink, especially when it can generate male population over the generation. So eventually, this entire population is going to shrink and get extinct. So this is being applied in controlling malaria and dengue, that means the vector-borne diseases. Not only that, it has tremendous amount of application in crop regulation, that means in the agriculture sector, to improve the production of crop or to develop plants which are capable of growing in hostile conditions. That means, for example, dry or very hot conditions, warm weather. The last one, which is very controversial, that means people have also applied CRISPR-based method in editing embryos. For example, in 2018, Chinese scientist Yang Kui, he edited embryo from two parents who are infected with HIV. Now he targeted the CCR5 gene, which is a receptor for HIV infection, and he was able to get rid of this gene. Now, if you do not have this gene in the embryo, then this embryo becomes immune to HIV. Then he's transplanted this edited embryo into the mother's womb, and she was able to give birth to two babies. Now, these babies are immune to HIV. So you can see the potential application and the the depth of the application and the broadness of the application of CRISPR in targeting various genetic and pathogenic diseases. The last couple of slides I'll spend on addressing whether we can also utilize human viruses. For example, now we have seen COVID-19, right? So these kind of viruses are extremely threatening to human population, right? Now the question is, can we utilize these kind of deadly viruses for the benefit of human? Can we target certain diseases? Yes, of course. For example, in this video, you can see people have used oncolytic viruses to target cancer in two different ways. These viruses, they can specifically target cancer cells and get into there and then burst it open and killing the cancer cell. At the same time, this cancer cell is going to release some antigens, which are going to be recognized by our immune cells. These immune cells are going to recognize the signal and then activate killer cells. For example, T cells, they can now target the residual cancer cells in the body and get rid of it. So people are also using viruses, human viruses, to target cancer, which are known as oncolytic viruses. One of the viruses already approved by FDA, which is known as Phage for melanoma. So with that, I hopefully I was able to give you how we can utilize threatening microbes, for example, both bacteria, bacteriophage, not bacteria, but bacteriophage to target bacterial infection. At the same time, infectious viruses to target different genetic diseases, for example, cancer. Now, at Ashoka, we are also working on the similar lines. For example, my lab at Ashoka, we are developing engineered microbes for drug discovery. So we engineer microbes or bacteria in such a way that they can not only synthesize active drug molecule, but also sense inactive drug molecule. That is how they can spit out active drug molecule and we can easily isolate them. At the same time, we are engineering viruses, human virus, so that we can increase the efficacy and targetability of cancer therapy. The third component that we are working on is looking at the phage resistance dynamics. So phage therapy has a drawback of resistance. In over time, these bacteria become resistant and then phage cannot act on them anymore. We're trying to understand how we can hinder the spontaneous point mutation based immunity in these pathogenic bacteria. At the same time, how can we block adaptive immunity in this pathogenic bacteria to increase the efficacy, and we are trying to understand the dynamics between all these players. Hopefully, this kind of research is going to improve the phage therapy efficacy, and that can be a potential alternate to the conventional antibiotic-based therapy. So with that, I'll conclude my talk here, and I cannot stop the presentation without thanking my group and also the funding agency. I thank Ashoka University for organizing this talk and UT for the coordinating the talk. Thank you all. Thank you all for your engagement. And if you have any questions, definitely you can reach out to me. And now I can also take some of the questions that you might have. Thank you.
Thank you, Vasudev, for such an enriching talk. We have a lot of questions pouring in. I'll start with the Q&A. So, Vasudev, the first question is regarding transforming the microorganisms into therapeutic molecule or vector. So, someone is asking, this may change the genetic constitution of microbes and may also cause immunomodulation, both harmful for society and human health. So should we use these microbes for the purpose of prophylaxis or treatment?
Sure. Now we need to understand that even though, let's say, if we do not deliberately change any genetic change, make any genetic change in these microbes, but there is constantly fight happening between these microbes, between bacteria and bacteriophages, and they are evolving. So you cannot stop their evolution. But what we can do, we can refine these changes, and then we can specifically use in disease context. Remember, we are not going to let these engineered bacteria or viruses or bacteriophages in the society or in the environment. So they are going to be used clinically, and they are going to be under high regulation. So there is no way that this kind of bacteriophages can come out into the society and then cause harmful, and then give rise to harmful effects. So that possibility is not there. But I would also like to add up to the concern that so scientists are also trying to develop molecules instead of instead of making the genetic changes in these microbes. What they're trying to do, they're trying to identify molecules which are responsible for the killing of these pathogenic bacteria. For example, some of these phages, they use a protein called lysine. So this lysine, they can lyse the bacterial cell wall and kill them. Then scientists ask, why can't we use the purified protein itself? So there is no genetic element involved, and there is no potential harm involved in this process. So people are also looking into that process, and people are trying to formulate the biomolecular medicines for targeting such kind of infectious diseases or infectious bacteria.
Sure. Thank you. So, Vasudev, the next question is, this is regarding antibiotic resistance. So what will happen if bacteria gets resistant to antibiotics? Does that mean we will have to create a new domain of antibiotics? If we create more antibiotics, won't that mean that eventually all the bacteria and other microbes will become more resistant? So what's the final solution? How do we deal with this problem?
See, we cannot stop evolution of organisms. They are going to evolve. Now, the question is, when we put them under selection pressure, especially when we expose them to the drug molecule, we have to make sure we kill them fully. There is a proverb called, which doesn't kill you, makes you stronger. The same thing is applied to the bacteria as well. If we apply medicine, for example, antibiotic drug molecule, which is not enough to kill them, then the bacterium is going to develop resistance, and that becomes deadly. The second time, this bacteria is not going to act or not going to respond to the disease, to the antibiotic drug molecule. So the first primary job that we need to do is making sure we are applying right dose of antibiotic and we are giving the full dose of the antibiotic to the patient. That is important. Otherwise, that can lead to the generation of antibiotic resistance. But that is only not going to solve the problem. There are other avenues, other domains as well. For example, recently we are seeing huge application of antibiotic drug molecule in farm, especially poultry farm, dairy farm, and all. And it's kind of like misuse. If you use so much of antibiotic and expose to environment, that's going to get into lakes, water, and different region, and there the microbe, that means the pathogenic bacteria which already present there, it's going to generate immunity. So it's going to get immune even before we expose them to drug molecule in our body. Let's say someone is infected with a bacteria. Okay, even though that person hasn't taken that drug molecule, you can find out that bacterium is resistant. Many of the times you'll hear the term MRSA, methicillin-resistant Staph aureus. So this is very common nowadays, becoming very common pathogenic bacteria in different hospitals. So this is happening primarily because of the misuse of and abuse of antibiotic and not applying antibiotic in the right formulation or right dose. So that's the reason. So to solve the problem, we need to come up with new drug molecule. Otherwise, we are going to lose the battle. So there will be upper hand for the bacteria, which you don't want.
Sure. Thank you, Vasudev. Now, next question is regarding CRISPR-Cas9. So, is it possible to edit genes using CRISPR-Cas9 technique in your lab? What are the different steps involved in the gene editing process using this technique, and is the process error-prone?
Right. So we do a lot of gene editing in our lab using CRISPR. Our primary focus is on developing new CRISPR molecule, new CRISPR protein, which can give you more precision, which can reduce the off-target effect. Okay, as you might remember that the specificity, it entirely depends on two components. One is the recognition, PAM recognition, the early sequence by the protein, that Cas9. The second element is the guide RNA. So the guide RNA has to be exactly same to the DNA type. Okay, now if there is a small amount of mismatch, then that guide RNA can target other region in the genome, and that leads to the off-target activity. So we are trying to understand if we can come up with better or more better active Cas9 with better precision. So our lab is actively working on developing CRISPR enzyme with more precision. At the same time, we are trying to regulate the activity using external stimuli, for example, drug molecule. We are trying to engineer the protein in such a way you can have control. You can control the enzyme activity from outside. Now you can activate and stop the activity of the enzyme whenever you want, and that can also reduce the prospect or possibility of off-target mediated genotoxicity. So yeah, we are actively working on those.
Thank you, Vasudev. Someone is asking, how can viruses kill cancer? So if you could again sort of share the process.
Right. So this was a serendipitous discovery. It happened in 1910 around, when a cancer patient got infected with viruses, and then doctors found that there is a sudden decrease in the cancer in the cancer state. That means cancer cells were kind of like shrink a little bit. Okay, so that made scientists curious whether a virus has anything to do with cancer, anti-cancer therapy. So now, as I said, these viruses are human viruses. That means they can target human. For example, coronavirus. Coronavirus can infect human epithelial cells, lung cells, right? That means it can get engaged with human cell surface and get into the cells. Okay, now once it gets into the cells, then what it does, it's going to hijack the cellular machinery. That means replication, transcription, and translation machinery, and then replicate itself. So it can form new viral particle inside the infected cell. Eventually, if the cell is overburdened, then the cell is going to burst open, and then cell is going to die. Now, the problem with normal human virus, they're not specific. Okay, they can target any cells, mostly the healthy cells, right? That's the reason scientists had to engineer this kind of virus. Okay, for example, the virus that is the Oncolytic virus that is approved by FDA, that is based on herpes virus. So scientists, what they did, they engineered this virus in such a way that they specifically can target cancer cells. They cannot touch the normal cells. This is happening. This can happen through targeting the cell receptor. So, for example, if you can present a protein molecule on the surface of virus, which can target the cell receptor specifically present on cancer cells, then what you can do, you can send this virus particle only to the cancer cells. So this is how the engineered Tvec virus was developed, and that is approved by FDA for oncolytic anti-cancer therapy.
Sure. Thank you. Vasudev, someone is asking, can a person get resistant to only a certain antibiotic and not to the other antibiotics? Is it possible?
Okay, that's a good question. So this kind of confusion is there in general public. First of all, you need to understand we don't get resistant to antibiotic. Rather, the bacterium that gets resistant. Okay. Now let's get into the details of the science. So one person can have different kind of bacteria, right? Now one bacteria can get resistant to one drug molecule. Okay. Now other bacterium can still survive in the person. That means that bacterium is not going to respond to that antibiotic drug molecule. So the antibiotic drug molecule makes the bacterium resistant, not the person. We don't get resistant. Okay, resistant. So the bacterium get resistant. Now, if you can clear up that resistant bacteria in some other way, then that's a better way of curing infection. That's another reason we need to have a parallel avenue, parallel therapeutic intervention. For example, utilizing bacteriophages. Now, if you can combine these antibiotic drug molecule with bacteriophages, then it can better target these infectious and multi-drug resistant bacteria. So the study that I have shown from ECSD, they took 10 patients which were multi-drug resistant. The drug were not responding. In that case, what they did, they combined drug molecule and bacteriophage together, and then they are able to cure 7 patients out of 10. So that means it can give synergistic activity. Okay, so the drug molecule is going to target some pathway, and there is bacteriophages, they are going to target another pathway to kill the bacteria. Okay, so it's a synergistic effect, and we can combine the activity to increase the efficacy to target this kind of infectious diseases.
Sure, thank you. So if you could share some other scientific uses of CRISPR-Cas9 technique beyond gene editing, are there any other sort of uses of this particular technique?
Right. So one thing I have mentioned that, so in the presentation, I have only talked about gene editing. The other avenue, the other domain that I haven't touched upon is the regulation, gene regulation. So one can, as you know, this CRISPR can recognize and bind to DNA in a sequence-specific manner. So what you can do, you can tag this CRISPR enzyme with other cargoes. For example, another enzyme or other drug molecule. Then you can site-specifically deliver these cargoes, for example, this enzyme and drug molecule, into that genomic locus. So you can very precisely deliver cargoes to genomic locus and modulate the epigenome regulation. That means you can regulate the epigenetic expression. So epigenetic regulation is also possible with CRISPR. For example, David Liu's base editing that specifically uses CRISPR enzyme, but it doesn't use nuclease activity. It uses nickase activity. That means it's going to target only one strand. At the same time, what he did, he fused other protein domains, which can perform precise chemical reaction on DNA. For example, he has fused deaminase, which can remove amine from DNA base, and that's going to lead to the mutation in the DNA. That means you can change one base to another very precisely without inducing a double stranded break. So those kind of applications are also possible. And another example is the diagnostic. So you guys must have heard about CRISPR, probably, Sherlock, to detect genetic elements, especially RNA. Now, from India, we have also developed, for example, it's known as Feluda, that came out from IGIB, okay, to detect DNA. So it uses another set of CRISPR enzyme called FN-Cas9, Cas9, and they're able to utilize this kind of CRISPR enzyme to detect DNA sequence. And it can be different kind of DNA sequence. It can be related to pathogen, it can be related to genetic disease, and so on and so forth. So you can utilize in molecular detection as well.
Sure. Thank you, Vasudev, for elaborating that bit. So considering the off-target effects, are there any ethical concerns of gene editing? And if we have the government's consent to use bacteriophages in human beings?
Right. So there are two parts. First one is the ethical concern with CRISPR. So far, CRISPR is not approved for application to apply in human beings. So it's still in the non-human primate stage. Okay. Now there is only one instance that came out from China where they applied CRISPR in editing embryo, which was not ethical because we don't know the off-target activity and the long-term effect. Okay, so that's the primary concern. As we do not have a comprehensive understanding of the off-target and long-term effect, that's the reason it's not approved yet for human application. There is another thing which has a serious ethical concern, that we might be able to edit somatic cells with CRISPR, but we should not touch the germline cell. Now, there is difference between germline and somatic cell, right? Somatic cells, they are already differentiated. They cannot give any, they cannot pass on to the next generation. However, germline cell, they can produce sperm and egg. And that means they can pass on the genetic element to the next generation. Now, if you touch the genetic material in germline cell, which is what's going to happen. Then you are going to pass that changed or altered genetic change, altered gene sequence in the subsequent generation, right? Which you don't want, because you do not know the long-term effect of this kind of genetic changes. So even it is approved, it's going to be approved only for the somatic cells, it's not going to be and should not be approved for the germline cells.
Sure. Thank you.
Oh, back to the second question.
The second part, right?
Yeah, bacteriophage. So it's to be honest, it is not systematically investigated yet. As you can see, this was the first investigation that happened in 2020 in ECSD, which is kind of like in an organized way, in the presence of regulatory board, ethical committee, and all. But the good thing about is, in this study, they are able to come up with positive outcome. That means it has prospect, but it needs to be done scientifically, and it needs to be done under regulation. So if we do that, and if we can improve the efficacy of bacteriophage therapy, is going to be in clinic in very soon. Not only that, it can be actually the patient can handle by themselves, because these are not going to be harmful. So patient can easily take these bacteriophage doses at home, and there won't be much cost or hospital related cost with this kind of therapy. So it's also going to take care of the hospital related cost as well, and that's going to help the many of the patients.
Sure. So next question is, if phage viruses can also kill good bacteria, also if there is a tendency that they can harm healthy cells or tissues of the target organism.
Right. So now we need to draw parallel between antibiotic and bacteriophage. One thing we need to remember, bacteriophages are going to be more specific than any of the antibiotic drug. Not only that, these bacteriophages are going to be so selective that it's going to target a very particular strain of bacteria. Okay, so it's going to be strain specific. That means it can target only subset of the bacteria. Okay, so it's not going to touch the healthy bacteria at all. Okay, so it's going to take care of the side effect. And the second part is whether it's going to affect the healthy cells. It should not, because it cannot infect human cells. It's going to target only the bacteria. That's the reason it should not affect healthy human cells.
Sure. So, Basudev, it was mentioned somewhere that viruses are being used to sort of cure sickle cell anemia in mice, etc. Is this sort of research being translated into human beings so far, or is it yet to be done?
No, it's yet to be done. It's still in the mouse model. So it's in the mouse model, then it's gonna progress very soon. But yeah, the research is happening very fast. In last 5 years, we have seen the development of the method to mouse model. Okay, so it has moved so much in this short duration. Now you can expect how fast it's going to move in the next couple of years. And if that gives proper efficacy, proper activity, then that's going to see the clinical component. That's going to see clinic as well.
Okay. So maybe one last question before we wrap up this session, as we are running out of time. So how do researchers sort of decide which viruses to use as phages, and what are the kind of characteristics that they look for, and how do they finalize the dose, as in how much microbes to be inserted to treat that sort of infection?
Right. So we have already a comprehensive understanding between understanding of the bacteriophages. That means which bacteriophages infect what kind of what kind of bacterial strain. So that data set is already there, but one can definitely evolve bacteriophage for a very specific strain through laboratory-based evolution. So you can make your own bacteriophage, which is going to be very specific to particular bacteria. So that is also possible. And these bacteria, the bacteriophage, they target receptor on the surface of targeting bacteria. For example, LPS, lipopolysaccharide. It can target flagella. It can target pili. And these are the receptors that these bacteriophages use to get into the bacteria and then infect them. Okay. And the dose. So the dose can be, that can be evaluated in lab-based biochemical acid, in cell-based assay. For example, you can use microplate to find out the efficacy of a dose of macrophages. So that is how even in UCSD or UCSD as well, they found out the efficacy of different strains of macrophages, and then tried to find out the suitable therapeutic window, and then applied that dose in the patient. Not only that, it was so safe, and they were so confident that it was done intravenously, IV. So it was in the bloodstream, and that showed quite impressive outcome, positive outcome.
Okay. Thank you so much, Vasudev, for elaborating these sort of powerful techniques and their applications. And this was a very informative session. Thank you so much for sparing time out for elaborating on all these aspects. So with this, we come to the end of today's session. And before leaving, we would request you all to please take a minute to share your feedback. The feedback form is now available in the chat box. The next session of Scientifically Speaking Season 4 is scheduled for 20th of September at 6 p.m. Indian Standard Time. The session will be delivered by Dr. Kalyan Chakraborty. He is the Assistant Professor of History at Ashoka University. The title of his talk is India's Oldest Meat Gravy and Milk, a peek into Harappan dining room. So this talk will be certainly at an interface of history, archaeology, and biology. I would like to inform our audience that the applications for undergraduate program starting in August 2023 will open next month on 17th of October. Please note that the registrations for the same are already open, and we would encourage students to register early and submit their applications for round 1 to get an early admission and financial aid decision without their class 12 board exams or common university entrance test. They will also receive personalized guidance and mentorship by our academic counselors to help you help you with the process. And Ashoka offers BSc honors degree in pure majors including physics, biology, chemistry, mathematics, computer sciences, as well as interdisciplinary majors in computer science and entrepreneurship and computer science and mathematics. With that, please do not forget to share your feedback on this session. This will help us improve for the subsequent sessions. And on that note, we come to the end of today's session. Thank you so much for joining us, and we look forward to your participation in the next session on 20th of September at 6 p.m. So till then, please do take care of yourself and stay safe. Thank you so much, Vasudev, for joining.
Thank you, and thanks everyone. Have a great weekend. Thank you.
Thank you. Bye, everyone.